Welcome to Unit 4, Lecture 7. So this is going to wrap up our lectures related with Chapter 5, and this one is all about enzymes. Now, we're going to talk a lot more about proteins in the next unit, but I wanted you to know that enzymes are a type of protein. And there's a lot of types of proteins that we're going to talk about, but that's the one thing is I want you to remember that enzymes are a type of protein, and they're a very good example of a protein. So think about the characteristics of enzymes and think about what happens with enzymes, and that'll help you with proteins when we talk about them in the next chapter, or the next unit, I should say. So enzymes, hey look, type of protein that usually end in ACE. Okay, they don't always end in ACE, but a lot of times they end in ASE. So remember, sugars, OS equals a carb, ACE equals an enzyme. So not all enzymes end in ACE, but if it ends in ACE, it is an enzyme. So enzymes are a type of catalyst. If you remember that from middle school, catalyst is something that speeds up a chemical reaction. So in this case, enzymes are known as organic catalysts because they're catalysts that are part of a living system. Organic, remember, carbon-based living system. Whereas you can also have chemical catalysts that are metals. You can have you can have a lot of different catalysts. Okay, so remember, as a catalyst, it works and it does its job, but it itself isn't changed. Okay, so if we look at down here, if we look at the process of a reaction, okay, during the course of this process, the enzyme is this big pink area. It doesn't change over the course of that chemical reaction. It stays exactly the same. What changes is the substrate. So we've got a couple of different things that are part of the enzyme structure. We've got an active site, which is where the chemical reaction happens. We've got a substrate, which is what the chemical reaction is working on. So in this case, maltose here, which is a carbohydrate, okay, is a substrate, okay? And then sometimes we have a cofactor or a coenzyme. This is another helping molecule. Okay, sometimes they're used, sometimes they're not. All right, and then over the course of an enzymatic reaction, maltose, you can see, fits into the active site. Okay, and then it breaks it apart into two separate glucoses. Okay, and usually what this glucose is going to go do is cell respiration. Okay, so this enzyme broke maltose apart that we can't use for cell respiration, broke it into glucoses, which we can use for cell respiration. So there are two, I would say, models. Okay, you've got the rigid structure. Okay, rigid structure basically means no movement. If you would, it doesn't change. The enzyme doesn't change. In an induced fit molecule, or an induced fit enzyme, it allows that enzyme to be a little more flexible with what the substrate is. So in this case, you can see it kind of has this little rigid active site here. And then once the substrate fits in there, the active site actually closes in around that substrate, releases the products, and then goes back to that original shape. So in this case, all right, this enzyme, while this may be a specific molecule, this enzyme could work on other molecules that are similar. So a rigid enzyme has one molecule it works on. Okay, one type, whereas an induced fit can generally work on a family of molecules where they can work on multiple things. So if we had another molecule that was a little more spread apart, the induced fit with that could work on that molecule as well. So generally with a rigid enzyme, one shape, one structure, one function. Induced fit, flexible shape, a variety of similar functions, okay? Enzymes can also be inhibited. What does it mean to inhibit? It means to stop, okay? So enzymes we don't always want enzymes to work, Okay, sometimes we want them to work, sometimes we want them to turn off, sometimes we want them to turn back on. Okay, and that can be for a variety of reasons. Some of it is natural. So some of it would happen on a regular basis just as the process of your metabolism. If you suddenly, let's say like pasta parties, okay? If you suddenly get an influx of glucose, of simple sugars, you don't need to break down fats or more complex sugars that you already have because you've got that influx of energy. Okay, so we would want an enzyme to turn off because we don't need to make more glucose because we've got it. In some cases, we want to turn enzymes off because they're overactive. And this is the process of a lot of drugs. A lot of medications will actually turn enzymes off because they're overactive doing something, or sometimes they'll turn them on because they're underactive, depending on what the situation is. But enzymes can be interrupted. Okay, and like I said, for good, bad, for lots of reasons. So enzymes can be inhibited when a chemical interrupts the function of the enzyme. And we have two methods. We have competitive inhibition, which is this guy right here. And then we have non-competitive inhibition. Okay, so competitive inhibition is something that binds to the active site and it stops the substrate from binding. Okay, so you can see here this molecule right here fit into the active site, and the substrate now can't get into the active site. So in this case, they are both competing for the active site. Whoever wins, wins. Okay, non-competitive inhibition, we end up having some other molecule that fits into another site, okay, on the enzyme. And what that does is, you can see it actually changed the active site, resulting in the fact that the substrate now can't bind. Either way, the substrate can't get to the active site, okay? But it depends on whether something is blocking it directly or something that is blocking it indirectly, okay? And like I said, this is where a lot of medications come into play. Okay, so wrap up. I want you to find the name of one enzyme that works in your cells and what its purpose. Okay, and then find the name of one enzyme inhibitor that can be used as a medication and what it does. All right, I will see you in class.